Hello, everyone, and welcome to today's webinar on Data to Discovery Unlocking MERSCOPE Ultra Insights with SpatialX, brought to you by Vizgen and BioTuring. I am Win Fang, and I will be your moderator for today's session. So before we begin, I would like to go over a few housekeeping items. So firstly, this webinar will run for approximately 60 minutes. You all are muted, but we encourage you to engage by submitting your questions in the Q&A box throughout the session, and we will address them during the Q&A. This webinar will be recorded, and you will receive the link after the webinar concluded. Without further ado, it's my pleasure to introduce you to the first speaker of today, Manisha Ray, the Director of Application Development at Vizgen. She earned her Ph.D. in biochemistry from UCSF and has remarkable background in applying new technologies to advanced scientific research, spanning proteomics, genomics, imaging, and bioinformatics, particularly in the study of cancer. She will be introducing the MERSCOPE Ultra, a platform that enables the mapping of transcripts across 3 centimeter square tissue on a single slide from a large variety of sample types. Over to you, Dr. Ray.
Hi, thank you for the introduction. I'll just share my slides. Can everybody see my screen okay?
Yes, we can all see.
Thank you for the introduction, and thank you everyone for joining us today. I'm Manisha Ray. I'm the Director of Applications at Vizgen, and I will be taking us through some overview and background of the MERSCOPE Ultra, which was used to generate the data that will then be gone into in more detail on the SpatialX platform by BioTuring. So to give a little background on Vizgen, Vizgen is based on the MERFISH technology, which was developed in the lab of Nguyet Dang at Harvard. This technology enabled spatial transcriptomics and was commercialized by Vizgen in 2021. So the first customers received it in 2021 with full commercialization in 2022. And the capabilities were further expanded in the subsequent years. So we've expanded from fresh frozen to FFPE, the ability to do protein co-detection. We have both custom panels and pre-designed panels. And in the past year, we had 2 very important new products that we launched. We expanded the capability of our panels to up to 1,000 genes, and we launched the MERSCOPE Ultra, which is the platform I'll be talking to you about today. And we are in the middle of launching our next generation chemistry, MERFISH 2.0. So over the years, Vizgen has enabled countless researchers to look at the spatial expression of their genes in a variety of applications. How the technology of MERFISH works is shown here. MERFISH stands for multiplexed error-robust fluorescent in situ hybridization, which is basically a very highly multiplexed version of single molecule FISH. So the ability to look at gene expression in spatial context. It works by assigning a binary barcode of 0s and 1s to each gene that you want to look at in your panel. The 0s and 1s correspond to fluorescent readout probes, and through multiple rounds of hybridization and imaging, we read out that series of 0s and 1s using the fluorescent probes. The barcode is then demultiplexed, and you end up with the gene expression, or the counts per gene, with XY coordinates, giving you spatial gene expression. This method has been rigorously validated in the field in a number of applications and areas. Again, the first paper was published in 2015, and since then, in the past approximate decade, well over 200 papers have been published in all of the best journals in many, many different subject areas. And the MERFISH and MERSCOPE technology are in use all over the world at top institutions across the globe. So it's a very, very well-vetted, well-established technology for doing spatial transcriptomics. Today, we're going to talk about the MERSCOPE Ultra, Vizgen's newest instrument for doing spatial transcriptomics using MERFISH. This is the second generation of our imaging platform and incorporates a lot of the feedback of the past 3 years on the market, and the MERSCOPE Ultra is available now. The platform itself is an end-to-end platform, starting from designing a gene panel all the way through analysis. You can design panels for your genes using our gene panel portal. We also have pre-designed panels, so we give researchers a lot of flexibility in terms of using an off-the-shelf panel or designing one specific to your experimental question. We provide researchers with a series of reagents and consumables for doing the sample prep, so fixing your tissue to a slide, embedding it in gel, clearing, and then adding the hybridization probes. And the actual imaging takes place on the MERSCOPE Ultra. So the multiple rounds of hybridization imaging happen on the actual instrument itself, which also comes with a dedicated analysis computer to analyze the large amounts of data that come out of each experiment. We also have some visualization tools to aid in the examination of the spatial gene expression. The MERSCOPE Ultra incorporates the 3 years of learning from our customers that the MERSCOPE Ultra has been out there, and it keeps all of the benefits of the MERSCOPE while expanding them for a series of additional benefits. So the MERSCOPE remains the only platform to commercialize and run the MERFISH technology, and MERFISH remains one of the highest resolution and highest sensitivity method to do spatial transcriptomics. We have the ability to look at up to 1,000 genes per experiment using both fully custom panels or pre-designed panels. And it's a very flexible system where you can look at a variety of different species and applications on the system. The MERSCOPE Ultra has all of those benefits, especially the high resolution and high sensitivity, but now has expanded capacity for imaging with a corresponding increase in computation and processing to account for that larger increase in imageable area. So the MERSCOPE Ultra now has 2 flow cells, a smaller and a larger flow cell, which enables you to image up to 3 centimeters squared per experiment. Because of the faster processing, you can measure up to 9 centimeters squared of data per week. The computation systems have been upgraded significantly, so experiments don't take 3 times as long to image. We've also introduced compression so that the samples and the files are easier to work with on the back end. And importantly, the MERSCOPE Ultra is backwards and forward compatible. So all existing MERSCOPE products can be run on the MERSCOPE Ultra, as well as future products such as the new chemistry, MERFISH 2.0. The real advantage of the Ultra is this expanded imageable area, which is enabled by the introduction of a second flow cell. The second flow cell is supported by a rectangular slide that you can use to section and place either a single larger tissue, such as a whole organ, or multiple smaller tissues, which will enable you to look at, say, multiple conditions in the same experiment. The slides themselves have been upgraded as well. So these slides now have markings on them to aid in the orientation and placement of tissues to make the most use of the area that you have for imaging. We also continue to have the smaller flow cell, which is uses a 40 millimeter round slide, the original MERSCOPE slide, which you can now use to image up to 1.25 centimeters squared per experiment. The smaller slide size means that you have smaller volumes, and thus you're using less reagent. So experiments using this smaller size will be faster and cheaper than when you're maximizing the imageable area. This makes it great for smaller tissues, smaller scale experiments, or for working out conditions before you scale up to maximizing all possible conditions. And both flow cells can be used interchangeably on the instrument, allowing researchers the maximum flexibility to meet their experimental needs. The ability to expand the imaging capacity by 3x will be transformational, but without a corresponding increase in processing, the data would quickly become very unwieldy. So the optics, the mechanics, and the computation were all upgraded to support this larger imageable area. So again, we can now image up to 9 centimeters squared per week. But the upgraded optics and mechanics enable that to happen faster so that it doesn't take 3 times as long to do a larger experiment. In addition, we've expanded the computation significantly to include a high fidelity image compression so that now the files can be made smaller, more portable, easier to use, and reduce storage. And the analysis is faster and more efficient so that the analysis will happen quickly and you can be ready to run your next experiment. In addition to the upgraded imaging, we have added additional storage capacity. So now each instrument can hold up to 58 terabytes of data, which means you can run over 10 experiments before you need to start moving data off the instrument. So you have a lot more flexibility in when and where you move your data. Importantly, all of these additional features were enabled without compromising the extremely high resolution and high sensitivity of the MERSCOPE. The MERSCOPE enables this high sensitivity and resolution through a combination of optics and the slides. Our optics have a very high numerical aperture. These very high numerical aperture oil immersion lenses, which enable very high resolution in combination with our slides, which are designed specifically for this high resolution imaging. What this means is the fluorescence from each individual spot that indicates the expression of the gene is as small and compact as possible so that you can collect a lot more spots per area. What this means is you get more data per cell, more counts of your gene per cell, which translates into higher sensitivity and more information about the biological question that you're studying. The outputs of the MERSCOPE Ultra enable a lot of different downstream analyses, which we will go into more detail with the SpatialX platform. But you get the list of all detected transcripts and their coordinates in a CSV and Parquet file. You get the actual images themselves in a TIFF file. You get the transcripts per cell matrix, the cell data matrix, and the cell boundaries in Parquet and CSV files. And you also get single cell clustering annotations as both a CSV or a H5AD file. All of these outputs are designed to be compatible with existing single cell analysis tools such as Seurat and ScanPy. So you can dig deeper into the data on your own or on our platform or using a platform like the SpatialX, which makes this a lot easier. So I'm going to talk a little bit now about the data that we ran for this experiment, which was then further analyzed using the BioTuring platform. And I want to note that this data was produced by Renqiao Chen, a scientist at Vizgen, who did all of the work presented here. So to take advantage of the larger imageable area capability of the MERSCOPE Ultra, we looked at neurodegeneration, specifically Alzheimer's disease, and sectioned a piece of normal human brain and a section of Alzheimer's disease affected human brain onto the same slide. So both sections were put onto a single large ultra slide and then run against a 1000-plex gene panel, as well as a protein marker for A beta, the hallmark of Alzheimer's disease. So in one experiment, we have both a healthy and diseased sample using the exact same panel, the exact same conditions, the exact same buffers, so we know there's no batch effects. And we were able to look at approximately 1,000 genes as well as protein, and then use that to do a variety of analyses. The panel itself was a 1000-plex panel. About half the panel was focused on cell typing, so identifying all of the different cells found in the brain. And the other half of the panel was focused on Alzheimer's disease. So there's many, many pathways associated with neurodegeneration, which you can see outlined here. So in addition to cell typing, we're able to look at a very comprehensive way of how the Alzheimer's disease tissue is affecting different pathways in the cell. This panel also covers many, many pathways common in neurodegeneration, so it's applicable not just to Alzheimer's disease, but to many neurodegenerative diseases. So here's what the data looked like at the end of the day. In the first panel, we're showing some common cell type markers, both in the normal tissue on the left and the Alzheimer's disease-affected tissue on the right. And you can see we see a very similar pattern of gene expression following the layers of the brain, which is what you'd expect When you look at the A-beta expression, again, as you'd expect, you see a lot more plaque expression in the Alzheimer's disease tissue on the right than on the left. And you can zoom into single cell level using the high resolution of the platform and really look closely at the interaction of the plaque with the surrounding cells and the surrounding tissue. And you can use that to do a variety of spatial analyses of what these plaques are doing to the surrounding neuronal tissue. And I will shortly pass this over to the BioTuring team, who will talk much more detail about how you can do very interesting analysis with this interesting experimental setup. So to wrap things up from my side, told you about the MERSCOPE Ultra, Vizgen's newest platform for doing spatial transcriptomics using MERFISH. It can image up to 3 centimeters squared per experiment. And we use this to look at both normal and Alzheimer's disease affected tissue in the same experiment. Vizgen is going to continue innovating and providing new products that further expand what you can do with spatial transcriptomics, including MERFISH 2.0, our new chemistry that will increase the sensitivity, especially in low-performing samples. And we'll soon be launching the ability to combine our pre-designed panels with a subset of custom genes to further increase the flexibility. So with that, I will turn it over to the BioTuring side so you can hear more about the very interesting data analysis you can do with this data on the SpatialX platform.
Thank you, Manisha. Thank you for sharing. Next, on to our next speaker, we have Nguyet Dang, a bioinformatician and product owner of BioTuring. She has extensive experience in multi-omic data analysis, specializing in developing tools and pipelines. She will be sharing on how SpatialX enables cell clustering, cell type prediction, differential expression, and regional segmentation.
Thank you for the introduction, and thank you, Manisha, for a very detailed explanation of the data and the technology. And hi, everyone, I'm Nguyet, and today it's my pleasure to show to you our platform SpatialX, and it can be used to analyze the MERSCOPE Ultra data. And we start with the agenda of my presentation. I will first providing the concept, the SpatialX, and then I will combine it with the demonstration to analyze the brain tissues from the MERSCOPE Ultra data, and then we left the remaining part for the discussions and the question if we have any. So the idea for the spatial data that we're having here is that we start to have different layers of the data available in one analysis. So we can have the expression matrix providing the gene expression levels for each cells, and it gives us the foundation for understanding the molecular landscape within the tissues. And then in the second layer, we can have the spatial coordinates for the cells and for the transcripts. And they are crucial because it gives us the information to map the physical location of each cells and transcript within the tissues. And then for the last layer, we can have the spatial image here where we can have the visual context of, and it helps us to interpret the data. And by combining all of these things, we will be able to provide a comprehensive framework for the spatial transcriptomic analysis. When we are looking at the data, it's come up to some requirements, like we want to combine all of the layer into a single analysis. And additionally, when the technologies evolves, we can have the data set with more and more data. It can come up to millions of transcripts or million cells. And the data resolution is increased also. And that's why it creates a blessing for the platform to have the capability to handle the submissions or the intuitive visualization for these three large components of the data set. However, when we are working on that project, we encounter some limitation that holding us back into the field. Like, for example, when we have multi-platform studies, it often lacks the standardization, and so it making it difficult to compare or combine the data. And moreover, the problem of aligning the tissue image with the sequencing data is difficult, and then to understand the structure of the data is challenging as well. So that's why we have to address the limitation in order to have the scientists to work with the spatial data and to unlock the potential of the analysis. And with that idea inside, for today's presentation, I would like to present to everyone the SpatialX platform. So this is one of the product from BioTuring that we want to use is to address the idea of the spatial data analysis, and we will provide the user the capability to work with the multi-image together, and we want to simplify the submission steps so that everyone will have the will be easy to submit the data and to perform the analysis. So how we are addressing it? Let me start the demonstration on the way we submit the data onto the platform. So here is the screen of the submissions data of SpatialX, where we can provide the part to submit different information here. So we have the image layer, the segmentation layer with the file at the Packet format, or we can also provide in the transcript and also the micro to mosaic pixel. So in this case, we can enter in the name of the sample or the data that we have. For example, I have a demo data here. So this is the sample names. And for example, in one sample, there might be the chance that we have different data set, like it can be the replicate of when we do the experiment. So they can stay put into the in this data set name here. So we have the demo data here. And in this case, we indicate that the platform that we use to generate the data in the Vizgen MERSCOPE data. And we have the right-hand side part here to provide the files that we want to use for the submissions. So you see that we have the detected transcript, the cell boundary, and also the image figure here. So when you provide the file in the corresponding folder structure here, SpatialX has the smart print option so that it can read through the folder and indicate which file going to which part of the submissions, and then you add the data set into the platform, and then you can click the submission. So after that, the process will automatically have you to submit the data into SpatialX. What happened when you click on the submissions button? So when we have all of the input from the data, we can provide three following steps to work on those files. One is the pre-processing step, where we're filtering cells that have no expressions. And then we will provide the option to run the embedding and the clustering here. So we will perform the SCPI embedding and perform the batch correction with Harmonize with the batch key is provided. And we also have the possibility to calculate the UMAP and the Disney embedding. And afterward, we will run the low-band clustering, a different resolution to initially generate the default visualization of the data for the users. And after that, the SpatialX will allow the user to access the figures for the three different data layers here. So user can see the spatial image, or user can see the cell center, the segmentation, and even the transcript location. And if we switch to the 2D mode, we can see the UMAP and Disney plot. So in this case, like Manisha has mentioned about the data that we have for the normal and the Alzheimer disease samples. And I would like to show everyone here how the sample look like on the SpatialX platform. Okay, so we come back here. So after the submissions, when we add two different files from two different samples, we are able to visualize the data side by side as the figure that I'm showing here to you on the platform. So on the left side, this is the samples from the Alzheimer disease, and on the right side, this is the sample for the have the normal condition. And what we are seeing here is the image layer of the data. And in this case, when you expand the image, you can see that this image was generated by having the data from different channels. Like for example, we have the data from the tau, the anti-p-tau protein, and also we have the amyloid beta at the green color here. So I can deselect some part of the image to see only one channel of the protein, and then I'm able to interact with the second image to show the information side by side. So in this case, when you are visualizing one single channel of the amyloid beta protein on the image, you can see that on the figures of the Alzheimer disease, you can see some amyloid beta block here, while in the sample of the normal one, you don't see much the block here. Yeah. So you will be able to zoom in or zoom out and interact with the image at the first layer here. Moreover, in spatial, we're also providing the cell and the transcript layer. So it will give you the possibility to access the segmentation information, and also it can give you the seeing of the cell center. For example, when you see here, we are adding up the segmentation information. And then if you want to see is you see the cell center, we can loading the information here also, that you see the information of the cell center. Yeah. And by using that, we also provide the option to access the third layer of the information, which is the transcript ones. So in this case, let's switch to the option of seeing a single image here. So in that single image, we have already loading the cell segmentation and the cell center here. So now we can perform the query gene on this sample to see how the expression look like. So in this case, I will search for the two genes that I'm interested in. So the first one is the SLC70A7. So this is one of the markers for the neural cells. And then the second one is the MOG genes that has mentioned in Manisha's slide. So, and then I will turn on the transcript level, and then I apply the search to see how the expressions look like in this case. So in this part, we can turn off the image layer to clearly see the distribution of the expression of the gene of these two genes in this figure, and you see that they are separating in two different layers here. And this gene, they are the marker used for different cell type in the in the brain. So in this case, you can use it to see how it's expressed, the location of the transcript across the brain sample. And then if you zoom in, you can see the expressions of the gene that like to see that if the transcript belonging to one cell or not. And in this part, I have providing a better zoom for a specific segment in this region. So you see that the gray layer here is the segmentation of the cell, and for this cell, it's mainly have the MOG gene, so it will be different from the cell beside that, where it's mainly express the SLC17A7 genes. So maybe these two cells coming from different cell type. Yeah. So the power of the SpatialX allow the user to interact differently with different layers of the data, and then it can help you to provide the insight for the samples at different conditions. Like you can see the image of the protein channel different across two conditions, or you can see the gene expression different between the two cells here. So when we see the pattern, we raise to the question that we want to understand, like which cell types are included in the samples, because we see a clearly different pattern from the gene expression and also from the protein channel here. So in this case, we will use a feature on spatial called the meta reference to help us answer this question, or the cell type annotation here. So what we are doing here is that initially using the gene expression profile, we are able to generate the cluster of the samples, and then using that cluster, we will compile it with the marker genes of the specific cell type to identify that each cluster they will belong to which type of the cell type. And at BioTuring, because we have a very big single cell database, so we have developed a feature called the meta reference. And in the meta reference, we have the list of the marker genes that corresponding to each cell type. So with that option, we will facilitate the task of the cell type annotation. So let me show you on the platform how we can work on that. So coming back to the figure here, we have an initial figures here. We can click on the analysis tab, and then the cell type prediction is in this part. And we can generate a new cell type prediction analysis. And in this case, we can select the reference, the single cell reference that we want to use to get the list of the marker genes. And because this is the brain sample, so we choose the we customize the database of the BioTuring, like we choose the one of the nervous system here, and then we generate the prediction. And it will take a little bit time to run on that, but here is a pre-run result for this analysis. So what you are seeing here is that when the analysis finished, you will see each cluster we has predict that, like how the percentage of the possibility that this cluster will belong to which cell type. So we have two groups here. Is the first one is the major cell type, and the second one is the subcell type. And you have the percentage of the cell type here. And for example, in some case, if you agree with the annotations, you can automatically use our automation and prediction here. But for example, in this first cluster, the Louvain cluster one, you see that we have the neuron in the subcell type, and we have other group here, like the GABAergic neuron here. So if you want to manually annotate the cell type, you can be able to do so. So we can click on the Louvain cluster one to get more insight to make a better decisions. And in this case, when we change to the Louvain cluster one, we can see that the list of the marker genes is available here. And there is some genes like the DLX1 or the CXCL14, they are belonging to, like they are important for the GABAergic neuron. So in this case, you can modify or manually re-annotate here. Like you can say that this one is the GABAergic neuron. And then you can go through different cluster to make the decision for the annotation. For example, in the Louvain cluster 4 here, the percentage, the actual size high, so you can still agree with our automation annotation there. And then after you go through all of the clusters, you can save the annotation to the metadata. So when you come back to the visualization here, we can see that there is the metadata of the cell type annotation that we are agree on. And then in this part, I can load the color of the cell type annotation on the figures. Yeah, and one important thing about the meta reference is that for the meta reference is that it can help you to facilitate the time and the work for you to easily do the annotation. And in our case, because we have the normal and the Alzheimer disease samples, so we have pre-run in advance the annotations for the two samples. So in this case, we combine the data of the Alzheimer disease sample and the normal sample at one layer. And then we compile the metadata of the cell type annotation for these two samples. And in this case, you can see that we have different cell type here. And if we want to see a specific cell type, like for example, the neuron, we have two types of the neuron here is the GABAergic neuron and the glutamatergic neuron. You can see that it is belonging to one region here. And for the other part, like we can have the oligodendrocyte and the oligodendrocyte precursor, they are more distributed in other part of the of other regions of the slide here. Yeah, so with the cell type annotation, we can answer the question, the first question, like what are the cell types that are available in the data set? But there's one thing that I would like to remind everyone here is that this answer is we are able to get it from the gene expression layer of the data only. But if we want to incorporate the spatial context into the analysis, how can we do that? And in this case, there are second features on the SpatialX that I would like to introduce at the moment is the region segmentation. The idea of the region segmentation is that beside the gene expression information, we incorporate the coordination of the transcript into the analysis, and then we will provide the region segmentation to see that which part of the image they are intend to have the similar expression profile. And in this case, let me run the analysis again. Okay. Here we come back to our figures. And for this part, the region segmentation button is available here. And then you click on that, we can run the new region segmentation. And in this case, we can define the radius, or we can define the clustering resolution. We can increase the resolution to get more cluster. And in the advanced parameter, it will ask us to indicate the Moroni threshold. For example, we can reduce the Moroni threshold, or we can increase the pairwise correlation threshold to increase the number of the segment that we want for the analysis, and then we can click the button run for the analysis. And it will take a little bit time to have the result available, but when you click on our pre-run results, it will load the segmentation result here. Okay. So in this part, what is the result presenting to us? So the idea of this result is that from the expression of a single image, we are able to identify 5 different regions based on the expression profile and its location. And then for each region, we are able to identify the marker gene representing for that segment as well. And you will be able to view the region one by one. So for example, when I click on the segment 0, you see that it is the purple color here. And then for when I move to the segment 1, they are concentrated in this part. And in this case, we have the list of the gene for the segment 0 and the segment 1, and we want to know that what is the pathway that are representative or enriched in the region. We can run the analysis like the pathway one here. And then initially, SpatialX will run the enrichment analysis for the gene that are able, the marker gene available in one segment, and then you will be able to perform the customized rerun here. So for example, if you in the rerun, if you are interested into a specific pathway like the disease database, for example, you can click on the rerun. And then what we are doing is that we perform the overrepresentation analysis, and then you can be able to view the result here, like the list of the enrichment ID that has been available by this gene set here. Yeah, and the idea of running the region segmentation after the cell type prediction is that we want to see that is there any similar pattern between the way that we use the gene expression only, or when we adding the spatial context, is there any different, or does the result they support each other? So in this case, when we're aligning the result of the region segmentation and the cell type annotation, we can see that they are they are supporting each other. Like for example, in this part for the oligodendrocyte, they are more belonging to the white matter, while the part of the neuron here. So here is the part of the neuron that we have seen in the cell type annotation part. It is more the neuron-rich region here. Yeah, so in this case, you can perform the analysis with the expression layer only, or when you incorporate the spatial context, you can see that the result of the region segmentation, they support each other. And then furthermore, if you want to investigate another layer of the spatial data, that you can come up with the next questions, like what are the differentially expressed genes between the normal and the Alzheimer disease samples? So in this case, the SpatialX can help you to answer this question as well. So let's coming back to this figure. So in this case, we have the differential expression feature here. And in my case, I want to run the analysis to see the differential expression between the neuron of the two conditions. One is the normal condition and another one is the disease condition. So in this case, what I'm doing is that first I choose the sample to compare is the normal one. And in this case, I select the metadata is that I will select the neuron. Okay, the GABAergic neuron. Okay, and then I clone this on the normal condition. And then for the second group, I will select the Alzheimer disease sample here. And then for the metadata, I will select the GABAergic neuron also. Okay, yeah, and then I call it the Alzheimer group. And then I can run the analysis of the DG analysis. And in one result that I already run here, so we will provide to the user the view that we have the table showing the log flow chains, and then also the FDR. And then we have the volcano plot here, and then the distribution of the gene. So when you have like two genes, the view of the expression, it will change to a volcano plot here. And in the case that I have searched the gene, the marker gene that are linked with the Alzheimer disease. So in this case, you see that the genes, they are differently expressed between the neurons of the Alzheimer sample, and when we compare with the neuron of the normal samples, they are expressed differently in two parts of the volcano plot here. Yeah, so in this case, we have been able to see that the SpatialX can help you to interact with your data at different layers. So you will be able to interact with the image, the cell information, and also the transcript layer. We provide the for the expression layer, we provide the analysis as in the single cell one, like you are able to perform the cell type annotation. And then for the spatial context, we adding up the region segmentation here. And finally, you can also run the differential gene expression to answer the questions of how your genes are expressed across different conditions for one cell type. And these are the features that I want to incorporate into this introduction of the SpatialX platform. Yeah. So in case that you have any questions or you want to discuss for any future features, I am very happy to answer it to you. And thank you everyone for listening. Yeah.
Thank you, Nguyet. Thank you for your presentation. Thank you, Manisha, for your insightful presentation as well. And I see that we received some great questions from our audience, and we will now move to the Q&A session. So there's one question from Anivan Paul, asking that is BioTuring going to implement a brain area segmentation step to annotate the mouse and human brain area? I think Nguyet can answer to this.
Yes, currently for the segmentation, it's work for any sample. So we you can submit the brain sample, and with the region segmentation, we can have to separate the segment in the samples. And then we're also working on the image segmentation. If you provide the image figure, we will have you to perform the segmentation on that also. So the idea is that we are not only limited to the brain samples, but the segmentation of the spatial, it can have to work on other sample as well.
Okay, and moving on to the next one would be, where does the data reside during the analysis? Whether it will be on your cloud, and what would be, whether there's a storage limit to hold the data, and if there's any additional cost implied to that.
Okay, so currently the SpatialX is a web applications, and the application will be deployed on the cloud storage. And depending on the type of the subscriptions, it depends on the way that the application is installed. Even it is on like BioTuring will host your application, or you will hosting it by yourself, the price for the holding the data or the storage limit will be different. So in this case, you can send us an email to support@bioturing.com to discuss in more detail about the type of the subscription or the cost structure for more details. Yeah.
All right. I do have a few questions as well before, and at the meantime, attendees, you also feel free to ask more questions while I'm giving up a couple to the speakers. There's one that I would like to know is whether this SpatialX platform, is it possible to integrate the molecular and spatial information for analysis? Like, for example, the molecular data is used to identify cell types, and spatial data reviews their distribution in regions. And can they perform like differential gene expression analysis within the same cell type, for example, oligodendrocytes?
Okay, so as we show everyone in the demonstration, you see that SpatialX will give the user the access to the expression level of the data, so you have the information of the molecular data. And moreover, with the cell segmentation and the cell center information, you can have the spatial information of where the location of the cells, and then with the transcript one, you will see where is the location of the transcript. And in terms of the analysis, the example that I have demonstrated at the end of the presentation is how we can compare the same cell type of different conditions, the normal and the Alzheimer's disease samples. And in the case that the user want to compare the same cell type, but in different regions, we can support the option to combine the metadata of the region segmentation and the cell type annotation into one, and then user will use that information to perform the analysis. So it is possible to do so and to have the user answer the questions. Yeah.
Okay. What about the output format? Is it compatible to use widely for single cell and spatial transcriptomic tools? For example, like ScanPy, ScriptPy, or further downstream analysis?
Yes, currently when you run the analysis and you want to export the data, for example, you want to export the expressions information, or you want to export the locations, it is possible to export the data at the H5AD format. So this is the one of the and data format that are supported by the ScanPy or the ScriptPy.
Okay.
Yeah.
Thank you, Nguyet. And the next one would be directing to Manisha. I would like to ask whether MERSCOPE Ultra can achieve single cell resolution in spatial imaging.
Yeah, so the MERSCOPE and MERSCOPE Ultra can get to single cell resolution. You could probably see in some of the data we showed today, where you can see the outline of each individual cell, the spaces between the cells, and each dot represents the location of that gene within the cell. So you truly can get to single cell resolution, and to some degree even single cell level resolution.
Okay, that's great. I think we have reached to the end of the Q&A sessions, and if there's no more questions, thank you everyone for joining us today and for your engaging participation. We hope everyone find this session valuable, and also special thanks to both of our speakers. Before you go, we would actually love to hear your thoughts about today's webinar. So after this webinar, you will receive a post-webinar email that consists of the survey as well as the recording links. So be sure to check your inbox for those, and your feedback will be very important for us to improve our future webinars. Thank you once again, and we look forward to seeing everyone in our future events. Thank you. Have a great day.
Thank you.